Hello, my name is Simran Gupta and I am a student in Mahi Institute of Technology. I am the student of Infection Risk Management Level 7. I am going to present my individual presentation on the microbiology of infection risk, and my topic is MRSA, which is also known as methicillin resistant Staphylococcus aureus. Now I am going to introduce my bacteria first. The introduction is MRSA, which is also known as methicillin resistant Staphylococcus aureus. It is a bacteria which resists from different actions of every antibiotics. This bacteria spreads from community, healthcare centers, hospitals, rest houses, and any other hospital equipments. Recent researches show that this disease is carried by a 50 million people worldwide. So it's a worldwide issue. These bacteria can be found on blood, skin, or the soft tissue of our human body. Now I am going to describe a structure of MRSA. The structure of MRSA is a bacterium which is a gram-positive bacteria. A mobile gene element called SCC which helps in the formation of this bacteria. You can see there is a FMM molecule components which help to provide them from penicillin resistant, which is the name of one antibiotic. You can see there scaffold protein which is known as collagen, lipids which are stereoxythene derived lipids, cargo which are multimeric protein complex. These are responsible for the strain treatment. Now the MCA gene, which is a gene code for PBP2, which is an essential factor for the resistance of antibiotics. This bacteria is categorized as a gram-positive and it is acquired as a genetic element, which is a mobile in nature. The name of the element is SCC-MEC, the size of which ranges from 23 to 64 kb. A gene fragment called MECA in this element has low harmony with PBP2. This enables MRSA to show resistance to essential antibiotics like methicillin. Now I am going to describe the virulence factor of MRSA in the human host. As we know, MRSA host is a humans which are very harmful. The harmfulness of MRSA in human host is which spreads from direct touch to form hospital equipments. Different parts of human host like bone, skin, tissues, soft skins and the delicate tissues which are get infected very easily. The virulence level of MRSA in human host depends on toxins. MRSA is very harmful to humans as it has immunity to a large range of antibiotics, making it. Now there is making it very tough to treat. However, recent research reveals that some low levels antibiotics can be used for treating the bacterial infection. I am going to give an example for this. For example, vancomycin is used for the treatment of MRSA. The virulence or the harmfulness of this bacteria depends on level of toxin. It can be seen that ligases like TSST ligase are held responsible for the launching of these toxins on human host. Now I am going to describe the mechanism of antimicrobial resistance in MRSA. As we see in the figure, it is the mechanism of antimicrobial resistance. In this, there is a presence of ribosomes which cause inactivation of phosphate group in antibiotics, and there are some drugs which get pumping out, and there is a modification which cause the target to the antibiotics. Now, what is their whole mechanism? I am going to explain this. Formation of biofilms which helps this bacteria to resist in the antimicrobial actions. Inactivation of various antibiotics is another process of resistance. PBP2A, which is a homologue of PB2A, which helps in the resistance of action. Biofilms are agglomerations which is implanted in the extracellular matrix of MRSA. Staphylococcus is considered as an excellent biofilm former which provides protection to the bacteria from the action of antibiotics. Moreover, enzymatic inactivation of antibiotics is another essential process which is done by the process of hydrolysis. Hydrolysis is the process done by in MRSA with the help of water, in which water helps in the breakdown of the MRSA protein. In addition, inactivation can be also achieved by creating inactive derivatives. These derivatives can be acquired from different category of resistant genes. Moreover, a genetic gain which codes for homologue of PBP2 gene. This homologue is also known as PB2PA gene. This gene can stop the action of drugs because it is located very deep inside. Now my next slide is on the immunological response mediated by MRSA during infection in the human host. As we all know that MRSA causes infection in the human host because it is resistant to many antibiotics. To explain this, we know that MRSA has the capability to destruct host defense which is mediated by their neutrophils. As we see in the diagram, the neutrophils which help the MRSA to binding up is which cause phagocytosis and the neutrophil get escape from the cell body. MRSA has the capability to destruct host defense which is mediated by neutrophils. Neutrophil action is stopped by phagocytosis as well as chemotaxis of the neutrophils. Moreover, it is argued by some researchers that Staphylococcus aureus has the capability to live within the neutrophils. It can be seen that the MRSA bacteria have the capability to innate immunity against neutrophils. This enables them to infect the human host by inactivating neutrophils. Staphylococcus aureus has the ability to destruct by initiating the action of phagocytosis. Moreover, chemotaxis of neutrophils can also be done by the bacteria. Chemotaxis process is achieved by the direct migration of the cells of neutrophils. Moreover, it is found from some researchers that this bacteria has the ability to survive within the neutrophils by the process of cytolysis. In this process, cytolysis local inflammation occurs in the host body. Now, I am going to describe the laboratory method of differentiation between MRSA and MSSA. Now, MRSA and MSSA. MRSA, we know that it's a methicillin resistant Staphylococcus aureus. So with the same MSSA, in this flow cytometry, we can see the differentiate between MRSA and MSSA. You can see the flow cytometry, there is a sample stain cells which get light and there is a laser light source which helps in the fluorescence from the stain cell, and these cells are getting forward and scattered with the help of light. It can be seen that flow symmetry paired broth cultures are used for this purpose. MRSA and MSSA is a differentiation is done on the basis of their count ratio, or we can also call this CI. Different laboratory methods are used to distinguish between MRSA and MSSA. The process of flow cytometry is done by using a micropro flow, which you can see in the diagram. Will you please push up the slide? In this diagram, you can see the flow cytometry, which helps in counting of microorganisms, pairing broth cultures by preparing using oxysaline. And labeling is done by using fasted total organism. However, this process of distinction consumes lots of time. This sort is continued by the flow cytometry. Now, you can see another test method for this, which is known as rapid slide latex agglutination test. This test is used to measure the distinguish between an antibiotics. In this method, we can collect the blood because blood collection is necessary for this test. MSSA is isolated by generating one PCR product, and MRSA isolates by generating two PCR products. So you can see in this diagram that one drop of positive control reagent is added to the first circle and one drop of latex reagent is dropped from the 5th and the 7th, which is a disposable test slide. After completing this method, the slide is rocked by hand for the 45 seconds and placed on a slide roaster for 45 seconds. After in the fourth process, there is a two complete chlorines of MRSA and MSSA. We can found it in an agent which are emulsified. In this test, we can measure the distinguish between antibodies. MRSA is isolated by generating one PCR product. MRSA isolates the generating two PCR products, as I told you previously. This is a screen test. This whole test is a screen test which is known as latent agglutination. Collection of blood from the patient needs to be done before conducting this test. This test is performed for measuring the presence of Staphylococcus aureus in the human body, in which MSSA separates by generating only one PCR product. However, MRSA is separated by generating the two PCR products. Now I am going to analyze the factors who are responsible for the development of genetically diverse MRSA. For to analyze this, CA-MRSA or the community acquired MRSA has increased due to low maintenance of personal hygiene. Unnecessary use of antibiotics has increased immunity of bacteria against those drugs. These drugs are stated that the poor maintenance of hospitals and hospital equipment has caused development for this. It can be seen that the genetically Staphylococcus aureus bacteria has become strong and evolved immunity against various drugs and antibiotics. This has happened due to unnecessary consumption of antibiotics without any proper prescription. Moreover, poor maintenance of hospitals has also initiated to the development of HA-MRSA. Now, there are risk factors which are associated with the MRSA from hospital. As per this diagram, you can see in the hospitals, the risk factors are prolonged hospital stay, often for the 14 days, receiving antimicrobial therapies, surgical procedures, presence in the intensive cares or burn unit, proximity of known which are known as MRSA cases. And in the LTCI factors, there are recent hospitalization, presence of decubit ulcers, indwelling catheter, high level of dependency. During the admission in hospital, MRSA can be acquired. Use of infected devices help in spreading the infection. Moreover, it is contracted that an individual having long-term treatment can acquire this infection. A person can be infected by this bacterium from any device in the hospital. Maybe it is catheter, sterilization syringes and all that. MRSA bacteria which is acquired from hospital is known as hospital-acquired MRSA. Using unsterilized devices like scalpels, scissors which can infect a person. Moreover, if a person is admitted to a hospital or a medical center for a long period of time, then the risk of infection increases. Now, I am going to conclude my topic with the conclusion. It is concluded that the several human practices have increased the ability of MRSA bacteria to become more stronger day by day, because these bacteria are getting resistance from many antibiotics and many drugs, and the MRSA in within few years may be proved deadly for the human beings or any other organisms. However, for the low level maintenance of MRSA antibiotics like vancomycin can be used for treating the different strains of these bacteria. Now, the scientist has proved that it can be made some new antibiotics to treat this MRSA. In addition, the awareness needs to be spread among community for protecting them from the harmful effect of these bacteria. Thank you for watching and listening me. This is Simran Gupta. Thank you.